Yeah, so good morning, everybody. I want to thank the organizers for the opportunity to present our work. And I have to say, it's my first time at the Oligo meeting. I hope it's the first out of many, because it's really a pleasure to be here and have a room with so much expertise for oligonucleotides. For me, this is a little bit almost like a humbling experience, because my background is actually in applied mathematics, and I work in a school of computing, and there I think about chemistry. So why would a mathematician in a school of computing think about chemistry? And if that's not enough, half of my time I also spend in a commercial position at Nanovary, R&D company in Newcastle upon Tyne, where I work in the biotech industry. And for me, the question that drives me mainly academically is, can we create algorithmic matter with material? So smart materials that can sense their environment, compute, process information from the environment, and ultimately act in response. And I wonder what kind of organizing principles and what kind of materials can we use to push the stream forward. And this has many applications, of course. Smart drugs is the one that's prominent here, but also in environmental monitoring. And so I foresee numerous applications for that. And so I wonder what drives that. And about 12 years ago, I stumbled into dynamic DNA nanotechnology as in what I see as an enabling factor for that. And because maybe people here are not so familiar with that, this is an introductory slide about what we call toehold-mediated DNA strand displacement. You see, for a mathematician, DNA, I don't worry too much about the exact structure. I don't even draw the double helix and just write complementary DNA as strands. They might have nucleotides, but I'm mainly concerned about domains on that DNA that have a certain complementarity, and I play with this complementarity information. And so if I take a double strand of DNA that has a short single-stranded overhang, which we call a toehold, typically 4 to 10 nucleotides long, then this can hybridize with an invading strand that has complementary region here, and you can have reversible hybridization between these two strands. If your invader has the same sequence than the upper strand of this double strand, then this can undergo a process called branch migration that goes randomly backwards and forwards until at some point this originally bound strand might be kicked out and you have an output. This is a reaction that has been proposed now 25 years ago by Jörg Simmel and Andrew Turbefield, and has pretty much created the field of DNA nanotechnology. For me, this is really interesting because it has been shown that, at least in theory, this allows you to model arbitrary chemical reaction networks. If we have some chemistry, for example, the Luzow-Jabotinsky reaction that can oscillate, or chemical reactions, chaotic reaction systems, or any complex kinetics, we can take this chemistry and transfer it one-to-one into a DNA strand displacement network, where you have strands. Here's a gate, for example, that can capture one strand, kick out another strand, thereby open a second toehold that can engage with a second input strand and kick out another strand. So this is essentially a model of some pen and paper chemistry that says A plus B goes to C plus D, and we can either have overhangs or additional domains, so that this D might go into a downstream reaction. And so you can start to plug and play together these reaction networks. We can design them really with pen and paper. So as a mathematician, I love it. Then we go to the computer. I engage with my students in the computing department to generate sequences that actually fold into this and optimize for the binding energies to get the kinetic constants, how we like them for our chemistry. And then we can go to the laboratory and actually observe these. So in theory, there is a proof by already by David Soloveichik from 2010 that with that we can model any arbitrary system. If I tell this to mathematicians, they're impressed. Chemists like you raise their eyebrow and say, "Show me." And so I show you a little bit of the pen and paper. So this is where this area of molecular computing then started, because the chemistry that I'm interested in is chemistry that we can interpret as performing some kind of computations. Has first been done by Selig and Eric Winfrey in 2007. And here's just some example of work. I don't want to go into detail with that. I just give you an overview of what I'm working there academically, where we want to embed these DSD logic circuits into DNA hydrogel that a collaborator of mine, Thomas Howard from the School of Biology, can print on glass support. So we can use essentially inkjet printers to deposit pixels of DNA hydrogel onto a glass surface. And we want to embed DNA logic gates that then function as a QR code generator that can sense environmental pathogenic DNA. And you could envision to put that into a shipping container with your produce. And when you open it at customs, you can directly scan this QR code and see whether there are pathogens in your load or not. This is kind of the vision. And we are currently in the side project optimizing the logic gates to get the most bang for the buck. We need as few gates as necessary in order to detect a growing number of pathogenic DNA by smartly reusing some of the components of the individual logic gates that we need. So this would be an example of what I mean by algorithmic matter. Can we do something that truly computes? If you want to compute logic is one good thing, but what we need is memory. And I don't mean memory in the form of DNA data storage, but some kind of dynamic memory like the random access memory in our computers. And so what I did at Newcastle University over the course of, I think, 5 years is to try to develop what we call in computer science a stacked data structure, which is a data structure where you can record information and then you can read it out again in a last in, first out manner, like a stack of papers. So we look into a data structure where you can push individual signals and then you can pop them out again and read what you had pushed. And you can go backwards and forwards in this process. And we essentially do this by growing a heteropolymer of little DNA. We call them bricks in a growing chain. And what we need to do is a controlled stepwise extension of this long strand that we have can store one signal after the other without runaway processes. And then we use strand displacement if we want to peel off the last recorded information so that we can read out what was the last signal that was stored, and then using either push-pop operations or push again, we can essentially dynamically increase or decrease the length of this polymer. I probably don't need to tell anyone here in this room that it's very hard to control stepwise extension of heteropolymers. In the end, DNA synthesis is all about that. And there you have different reaction environments. But we had to do this all in the same reaction environment. So in order to control runaway processes, it's very crucial that at the end of every single operation, you wash out any remaining supernatant signal, because if there is some signal that you wanted to record one instance of, if this is left in solution, and you put your pop signal, your pop command into the mix, then this will just run off and you have an extensive polymerization of many signals. So we really want to, and my student, Nunzia Lopiccolo, she fine-tuned a lot of washing protocols in order to separate our growing stack from the operating strands that we used here. And this has been published already 5 years ago in Nature Communications, where here you see essentially this is capillary gel electrophoresis, and here is page electrophoresis of our growing DNA strand, where we have recorded 1, 2, 3 signals. All right. So after fine-tuning this protocol a lot, we did manage that we can iteratively read and record and record, read out, and re-record several bits of information. But what you also can't help noticing, I guess, is that there are multiple side bands of undesired side reactions, where we see that even if we wanted to record only 2 signals, we still see a band of 3 recorded signals. And if we read out after the last recording, we get the majority of the populations of our stacks in the correct state, but some of them have just read out more than we wanted. So it's a very tricky protocol. And so what we decided is to develop a rule-based model of this chemistry, where we can follow individual populations of these growing stack components. And every time you would push something, if there is in the model some signal left, then it correctly models this runaway side reaction and things. So we did not constrain the state space of this model, but let it explore what could actually happen. And then we implemented our chemistry as well as the protocol that we are using, and tried to found what are the most important parameters. And it turned out that that the quality of the washing is really what constrains us to not go further than a few bits. So this would not scale to megabyte of information that we can store like this. So we don't compete with DNA data storage. We want this operative memory that we can short-term store some signals. This work that is now, I don't want to optimize this anymore. There were 3 major conclusions that I had out of this work. For me, the first one was that maybe going into following this approach that we know from computer science, that we need logic gates and memory, it's not the right approach if we want to do computation in chemistry. And so I started to look into neural networks, because it turns out that their elements, individual nodes in a neural network, we can just as well easily capture using strand displacement reactions. This was pioneered by Lulukian in 2011, and there are a couple of very interesting papers where they go all the way to do handwriting recognition using strand displacement reaction cascades. So there's some existing work on that. And currently I have my PhD student, Ethan Collinson. He did a very large literature review to investigate and model all different motifs people have suggested to build molecular multipliers and molecular rectifiers, which are the basic components you need to build a neural network. Then he compared their fidelity, how closely do they work together with a against the specification of the neural network that you would like to have, and also how compatible they are. Can you combine a seesaw multiplier with a decoy rectifier? Yes, that works, but if you use another rectifier motive, they are incompatible. And so he has a whole, was able to generate a whole matrix of which of these motives work together with which other motives. And now he is condensing this learning outcome into an automatic pipeline where you can convert a computational neural network into a DSD strand displacement network that you can then essentially order straight away. So if you want to, you could from sequencing experiments, you could get biomarker profiles, and then on your computer you can train a computational neural network, and then using the software you convert that into a chemical neural network that you can then directly apply to detect your biomarker signature in a sample. So this was one outcome on the academic side. I'm quite excited to go further into these neural networks. My PhD student, Jerzy Kozira, his outcome was that he really liked DNA nanotechnology, and he went on 7 years ago to found the company Nanovary, which now offers DNA strand displacement products in the commercial context. And my third learning outcome of this project was that rather than me supervising chemists as a mathematician, I should really hook up with expert chemists. And so I also joined Nanovary part-time, where we have our research director is an extremely skilled polymer scientist. And so we developed commercial DNA strand displacement system, particularly for the detection of modified oligonucleotides and oligonucleotide therapeutics. So if you're a PhD student optimizing hybridization analyzer, then you might be familiar with these common problems in bioanalysis. You have complex reactions and protocols. You have an interfering matrix. Your tissue throws off your experiments. You have chemical modifications and conjugates that might impact your detection abilities. And so what we developed over the last years was simplified quantification protocol and an assay for oligonucleotide therapeutics. Essentially, it's a two-step protocol where you have your sample, you add our nanorobots, and then you put them in a plate reader and measure the fluorescence. And so this protocol is tailored to ASOs and small interfering RNAs present in your sample. We then detect this RNA, amplify the signal to ultimately get a fluorescent response from it that you can read in a standard plate reader. It works directly in your biological samples. It's enzyme-free, which makes it really robust. It's isothermal. You don't need even a qPCR machine, and it's very simple to use using standard tools, which are very widespread, and it's cost-effective and simple. But the mechanism of action behind it is essentially catalytic amplification reaction. Since I made you all familiar with strand displacement reactions, we have our small DNA nanorobot that can capture your target sequence, displace a strand that is fluorescently labeled, and you get an increase in fluorescence. But because we want to measure trace amounts of your target, it's not enough to dequench one fluorophore per molecule. So instead, we provide a fuel molecule that kicks out your sample, your target again, so that it can engage with a nanorobot and do this over and over. And so what you get is kinetic fluorescence traces, and that should be an animation. Yeah. So what you get if you put it in the plate reader, that you have kinetic traces. I don't know how to do this animation. It doesn't matter. I show some static. We just. Oh yeah, thank you. What would I do without you? so essentially, depending on your input concentration, you get rising traces of fluorescence, and the reaction rate is proportional to your concentration. And what you can see here is an assay that goes from the tens of picomolar up to nanomolar, more or less. We have developed this assay to work for ASOs, siRNAs. We're working on aptamer detection and engage in some programs here, microRNA, you name it. Works with all common chemical modifications, PS, LNA, OME, MOE, so that the standard substance, and I would dare to, even if you have an unconventional modification, let's try it as long as it can stick nicely to DNA. I think we're happy to detect it. We could show that it's compatible with conjugates on the 5' or 3' and small molecules or large proteins or antibodies. They don't throw off our detectors. And you can use it directly in biological matrices, such as serum, plasma, or tissue homogenates. Works in a high throughput scenario. We have a dynamic range over 2 orders of magnitude and quite a nice sensitivity in the dozen of picomolar range. So here's just showing you our sensitivity for all different, for many different modified ASOs. And you see that our response curves pretty much overlap independent of the decorations or the modifications you do. Works for ASOs and small interfering is just the same. What we essentially do is, if our target is double-stranded, by mixing your liquids, you change the reaction environment such there is a pH switch that opens your double strand to make either the active or the passive strand accessible to our technology. As I mentioned, NANs can operate in a wide range of environments. So whether it is water, serum, or plasma, you essentially get the same response. And also, if you work with tissue homogenates, we could detect ASOs in brain, plasma, and even in liver, which is a little harder. The exact dilution depends on the tissue. Typically, if we work in plasma or serum, you have a 1-to-1 dilution, and for more complex tissues, it's 1-to-9. All right. We can design nanorobots to be very specific, sequence specific. So here's an example where we developed a nanorobot that is triggered by LED7A, but not by any other microRNA of the same family, even though they differ only in individual point mutations. Maybe we can even multiplex this and develop multiple nanorobots and let them run simultaneously in the same sample, and they give their responses using different freedom dyes. And just to show you a few case studies, this has been validated against ICH-M10 guidelines to ensure accuracy across different laboratories. So here was a recently published study on our side and with AstraZeneca. And you see that the limit of quantification is the same over the 2 sites. Here's a study together with NATA down here in Oxford, done by Talentari, where we look into MOE gap mares and PK studies in different mouse models. And what you can see is that it works for an MOE gapmer, for an ABC DNA gapmer, and also for a conjugated ABC DNA gapmer. And we do find uptake in the mouse tissue well, we can measure that in all different samples. Brain tissue study that we recently did, where we can show you that with tissue homogenization, we can detect the different levels of the target aptamer in the tissue directly in the samples. Maybe I skip this slide. If you're interested into data analysis, and talk to me in the break, but just in the interest of time, I come to a close. So if you develop PK studies, then please talk to me, because we have a solution that works directly in biological media, no sample extraction required. It's fast and has a simple workflow. It's built directly for ASOs and siRNAs, but if you work with aptamers, also come to talk to me. And it suits a wide variety of chemical modifications and conjugates. And with that, I just want to acknowledge collaborators in the School of Computing, Nunzia and Jurek. I pointed out for the stack work, Thomas Howard for the DNA logic. And at Nanovery, I want to mainly point out Alex Jackson, who is our research director and a very skilled DNA chemist. And with that, I open for your questions.